start?
Good morning, everyone. Today we will be talking about CRISPR-Cas9 as a potential solution for Huntington's disease. By Albert, Elena, David, and myself, Luke.
Huntington's disease is a rare inherited neurological disease that breaks down nerve cells or neurons in the brain that control voluntary movement, and it progresses over time, meaning the first symptoms are typically observed in middle-aged people.
So as Elena said, Huntington's disease is a neurological disease that causes involuntary movement and affects parts of the basal ganglia and its related structures, which help with motor control and functions with movement. And so this happens because of amyloid aggregates, as you can see under D. It attacks the nerve cell, which disrupts nerve function. And the reason that happens is because the HTT proteins from the HTT gene create amyloid aggregates.
The disease is inherited through a mutation in the Huntington gene, which causes more CAG, cytosine, adenine, and guanine repeats than usual. As you can see on the figure on the left, there are more CAG repeats in the mutation than in the normal Huntington gene. And the table below the text shows that 10 to 26 repeats is considered normal, and while 27 to 35 repeats means that the person won't have HD, they still have a chance of carrying it on to their offspring.
Although Huntington's disease cannot be cured, it can be treated and managed with drugs such as haloperidol and tetrabenazine, which helps with the unusual movements caused by the Huntington's disease. It can also help treat the hallucinations and delusional thoughts caused by this disease. The drug works by attacking the mRNA, which sends the damaged protein, or which creates the damaged protein. So what the drug does is it attaches to the mRNA and prevents it from creating those damaged proteins. And there are alternative methods such as gene altering, but there is no guarantee that it'll work.
The most common and efficient way to diagnose Huntington's is through a direct genetic test to count the number of CAG repeats. And as said before, if you find 36 or more repeats, it supports the diagnosis of Huntington's. But if you have less than that, you should be good for the most part. And then another way is to take a neurological exam with the doctor or a medical professional that you can schedule with. And just to get a heads up to your susceptibility of Huntington's is through an examination of your family medical history.
So a potential solution for Huntington's disease is CRISPR-Cas9. CRISPR stands for clustered regularly interspaced short palindromic repeats, which is basically a defense mechanism against bacteriophages, so like illnesses and stuff like that. And Cas9 is an enzyme that actually does the cleaving to the targeted genome. It comes from Streptococcus pyogenes, I think. And so yeah. So this is CRISPR-Cas9 in action. So the first thing CRISPR needs to actually cut the targeted gene is a matching PAM sequence, and PAM stands for protospacer adjacent motif. And what it does is it needs a matching PAM sequence. So in the mutant chromosome, you can see the NGG, which is just a nucleotide base, where N stands for any type of base, so it can be guanine, cytosine, or thymine. And so what it does basically in the mutant chromosome, once it finds the matching PAM sequence, it basically goes a little more upstream, and then it makes the cut on both sides of the DNA string.
CRISPR was first discovered in the sequences of the DNA of E. coli in 1987 by Ishino et al. In Osaka University in Japan. And then as time progressed, the research and technology also progressed with the most recent addition by Jennifer Doudna and Emmanuel Charpentier, where they really found out how to extract the sequences of DNA containing CRISPR from the E. coli using molecular scissors. And this is really the process from start to finish. As soon as the CRISPR is injected into the organism with the unwanted gene. So first the guide RNA, or gRNA, identifies the target sequence, and then it takes Cas9 to the target sequence and begins by attaching to the target sequence. And then it's removed, and then depending on the goal, the empty space is either left to be repaired by the DNA or it's substituted by a desired gene.
So
So there are pros and cons to using CRISPR technology. Some pros being that gene editing in CRISPR is relatively inexpensive in comparison to other gene editing methods of this time. There's also the ability to quickly create cell and animal models which can be used to research mental illness and cancer. And it could eventually be used to treat diseases that could only be treated through edits of the genome. Some cons are that CRISPR might be used unethically for creating designer babies or super soldiers, and there's a risk of CRISPR making a mistake and cutting the wrong gene, which would lead to a chain reaction and affect the entire cell.
CRISPR technology is currently only being used in clinical trials. Some clinical trials are charging anywhere between $1 to $3 million, and others are charging anywhere between $10,000 and $30,000. Over time, with the continuing research of CRISPR, the cost of this biotechnology has been decreasing over time, shown by this graph. And this should allow more people to be able to access technology in the future. Thank you.